Good morning, everyone. So first of all, I would like to thank the organizers who has organized this beautiful event, and there is so much scientific stimulation going on, and they have given me this opportunity to talk about my research, which I have been carrying out in Professor Doug Weibel's laboratory in Department of Biochemistry. I'm in my 5th year of graduate studies, and I have been working on multiple projects, as Nick talked about, like how mechanical anisotropy of the environment affect the bacteria motion, their behavior. Recently, I started this project in which I'm studying how biomechanical regulation from the genome of bacteria governs its properties, mainly mechanical properties. So in our laboratory, we have developed many tools to study the mechanics of bacteria, and these observations, which I'm going to show you, will help us understanding new biochemical pathways and hopefully come up with novel targets to treat infections and bacterial contamination. So just to give you some overview why bacterial contamination is a big deal. So in nature we have so many bacteria. Some of them are good and some of them are really bad boys. So they are ubiquitous. They cause so many infections, and they are contaminants in many industries, especially healthcare, food industry, water treatment plants. And they usually form biofilms, or people call it slime, and it gets really difficult to treat these kind of infection because biofilms act as a protective layer. So even if you treat these biofilms with bunch of antibiotics, it's really hard to penetrate the biofilm and treat the infection. On top of that, these bacteria are getting resistant day by day. So people have estimated the cost, annual cost, due to these contamination, and it is more than 1 trillion. And these estimates are coming from CDC and FDA, as well as from different journals from food industry and marine industry. So what we are trying to do in Weibel Lab is to develop novel antimicrobial compounds against genes, which we have found in this study, as well as we are trying to develop a research program in our lab to come up with really cool designs of small molecules to inhibit these contaminations as well as infections. So let's think about bacteria. So bacteria come in all sorts of shapes. They are rod-shaped, they are spherical, some of them are star-shaped, and they have flagella. Some of them, they don't have flagella. They sometimes form these long chains in the environment. But people have been studying how come these bacteria come up with these interesting shapes. And the theme which we have found is their envelope is very different in these different shaped bacteria. And broadly, biologists have grouped these different shape of bacteria into 2 groups based on their envelope properties. So some bacteria are gram positive and some of them are gram negative. And if we look at the membranes or the cell envelope of these 2 different types of bacteria, gram negative have 2 layers. One is outer membrane layer and the other one is inner membrane. In between these 2 layers, they have peptidoglycan, which is a polymer material in these bacteria. The other kinds of bacteria are gram positive. They don't have inner membrane layer, but they have very thick peptidoglycan. And so there has been studies to model this biopolymer, which is very thin in gram negative and very thick in gram positive. And these models suggest that this polymer material is not homogeneous. There are multiple holes through which these antibiotics can penetrate in. And if we look at the structure of these biopolymers, they are made up of these glycan strands, which are in green. And these glycan strands are connected with peptide bonds, which have been shown here in red. So depending on the cross-linking of these glycan strands, bacteria become very rigid or very soft. And the rigidity and the softness of bacteria gives it, an advantage to modify according to the environmental pressure, and also it helps them to combat the antibiotic pressure we apply. So just to give you an example, in this particular image, you you can notice that one bacteria is dividing and it becomes 4 after 2 rounds of division, and the only difference in these images is this bacteria has less cross-linking in these glycan strands. So if you have less cross-linking, over time, after 50 minutes, these bacteria explode itself because the bacteria has this cytosol, which is containing a lot of proteins, DNA. Over time, if you don't have a really nice stress-bearing material surrounding the bacteria explodes. In this particular movie, so what I'm showing in this movie is the cells are dividing, they are getting longer and longer, and they are popping because the bacteria cannot keep up with the osmotic pressure it is building from inside due to the synthesis of proteins, due to the replication of the DNA, due to the formation of multiple oligopolymers within the bacteria cell. So what we have come up is 2 tools. The one tool which I have shown here is clamp. So what we do in these methods, we take the bacteria cell, we encapsulate this bacteria in a polymer, agarose, and depending on the concentration of agarose, we get different stiffness of the agarose gel. And if we grow the bacteria in these different stiffness of agarose gel, we see a change in the growth rate. So higher the agarose concentration, the growth rate is lower. And you can imagine with this example, so suppose I am a bacteria and this is agarose environment. In order to grow, in order to divide, I need to push this environment. And I need to be very strong to push this environment. If I am not strong enough, I won't be able to push, I won't be able to divide. So the bacteria which are really strong will be able to grow. The bacteria which are softer will not be able to grow. So this methodology helps us understanding how bacteria can divide in stiff environment. And depending on the growth rate, we can compare bacteria from one species to the other. Within one species, based on their growth rate, we can also compare their pathogenic property, which I will talk about in next couple of slides. The other methodology we have developed is based on microfluidic device. So in this device, as you can see, there are multiple channels. This is a side view. So what we do here, we apply some vacuum suction to load this device with cells. So initially we flow cells in these microfluidic devices. We apply some suction, the bacteria gets inserted in these channels, and then we flow different medium. Depending on the flow rate, these bacteria bend, and we calculate the bending rigidity of these bacteria using this methodology. So why we need to identify proteins in E. coli? The reason is we have been using the same antibiotics which have been discovered decades ago, and we are just modifying their functional groups. But we are not coming up with innovative design or innovative targets. So this methodology or this project helps us discovering new proteins from genome-wide studies in E. coli. And our approach is to systematically delete each gene and then measure the stiffness and then identify key proteins. So for an example, if the bacteria has 20 genes, if I delete gene 1, I will measure the stiffness. If this gene is responsible for the stiffness, upon deletion, the bacteria will be softer. If this gene has nothing to do with the stiffness, the stiffness will not change. And then once we have identified these key proteins, we can come up with inhibitors against those proteins which make the bacteria softer or stiffer. So the E. coli genome, if we think about it, it is 4.6 gigabases. And this is a circular map of E. coli genome. And in this map, there are 4,500 genes are shown. And what we have done here is we have gotten a library of these E. coli bacteria in which one gene is deleted at a time. So out of these 4,500 mutants, we have individual strain collection, and what we are doing in our experiments, using one mutation at a time and then doing high throughput mechanics study to find out if this gene is responsible for the stiffness. And this methodology of encapsulating bacteria in a clamp is high throughput, so we can screen bunch of genes at a time, and there is no tools available before this study came to calculate the stiffness. So here I'm showing you the whole methodology, how it works. So here you can see the 96 well plate. We divide this plate into two halves. One half we use agarose solution and we grow bacteria in it. So initially we put agarose, we add some cells, and then we use magnets to mix the bacteria with the agarose solution, and then we let it grow overnight. On other half of this plate we have bacteria growing in LB medium. And in the end, what we calculate is GRAB score. GRAB stand for General Regulators Affecting Bacterial Stiffness. And these GRAB scores are normalized against agarose growth as well as LB growth for mutant as well as for wild type. And once we get these GRAB score, we can compare the stiffness of bacteria and their mutants. So for an example, there are 4 scenarios that can happen. So look at, if you look at this panel, we have wild type bacteria which grows as shown in dotted line in LB versus agarose. So you can imagine in agarose, bacteria needs to push this environment, so the growth will be slower. But in LB, it is happy to grow and the growth is way higher than in agarose. So now, using this wild type bacteria, if we grow mutation of a gene, then in the green it is shown that if the GRAB score is 0, then the mutant will grow as fine as wild type in LB as well as in agarose. So mutation doesn't affect the growth of bacteria, either you are growing in wild type or in LB, or in agarose or in LB. Now, if the GRAB score is 0, meaning that bacteria is becoming softer upon mutation, so the mutant is growing as fine as wild type in LB, if you compare blue and dotted black, but it is growing less in agarose compared to wild type. So meaning that this bacteria cannot push the environment in order to grow, and this gives us less than 0 GRAB score. Now, there is another scenario in which bacteria become more stiffer, and in that case, bacteria is growing fine compared to wild type in agarose, but it grows very less in LB. So by calculating GRAB score from these 4 scenarios, we can compare the genome of E. coli. And here we have plotted a scattered plot for the growth of bacteria in agarose as well as in LB. And I have highlighted this is the wild type. So wild type grows really well in LB as well as in agarose. And in this black circle, we are seeing a lot of genes. These genes are different than wild type, but we think that this difference is not significant enough. In red, I am showing one gene that is growing okay in LB, but it grows really less in agarose, meaning that this gene makes the bacteria stronger if this gene is present in the genome. And if we delete this gene, bacteria becomes softer. So people have been thinking that genes which are related to cell wall synthesis and metabolism are the only genes that make the bacteria strong. But what we have found using this whole genome screening methodology, that there are genes which have functions like replication and recombination pair, energy production, which has not been envisioned before, that genes doing entirely different function are contributing to stiffness. So our study has shown that genes with very different function also contribute to stiffness. And here I am showing you a frequency plot with multiple genes with their GRAB score. Most of the genes are concentrated at the GRAB score 0, but there are some genes like this MRCB, which is way down here, and there are some genes like REC-A or ACEF, which are way up here. Now, I'm going to show you how these microfluidic based assay confirms the reduced stiffness of mutants. So as I told you, in these devices we have bacteria captured in these microvels, and we are flowing this medium. And what we are measuring is the bending rigidity based on this formula. E is the Young modulus and I is the second moment of inertia. By calculating the force, length, and the deflection, X max, we can come up with these parameters, bending rigidity and second moment of inertia. And what we found that in these microfluidic based assays, we get the same result as our clamp assay. So for an example here, this is a representation of how our data looks like. So here we have wild type, and this is in the blue we are seeing the cells captured in the device. And here we have one example of a mutation MRCB. Cells are standing straight, but as we flow the medium, these cells bend, and we calculate the bending rigidity from mutant as well as from wild type, and then we plot here. As I told you, MRCB is one of the gene we found contributes to stiffness from clamp screen, is also gives us the least bending rigidity from our bending device experiments. And the gene I mentioned, REC-A, is giving us higher bending rigidity modulus, which is the same as we got from our clamp data. So what we have found here is these two tools can be used to study any bacteria, any organism, to find what are the genetic modulators that control the stiffness of bacteria. And we can use these tools to come up with novel inhibitors. So what we are doing here is we are doing high throughput biophysics to identify proteins. These proteins can be targeted using novel small molecule inhibitors, and these novel small molecules inhibitor can be used to disrupt cell wall assembly. So the conclusion of my results are these genome-wide identification of cell mechanics modulators will help us understanding the physiology of bacteria. It will help us finding new proteins, new pathways that contribute to the stiffness. These proteins can be targeted using chemical biology approach. We can combine the mutation of two genes, and then we can get an enhanced phenotype. And also we can do some biopolymer engineering. As I showed you, if we change the cross-linking of these materials, the bacteria explodes. So we can design new materials based on expressing or down-regulating these genes. And we can also make more cross-linked polymer or less cross-linked biopolymer. With this, I would like to thank my professor, Doug Weibel, and lab. These are the funding sources for our lab. Thank you so much.
Do we have time for a question or two? You mentioned that you identified genes which affect cell stiffness that previously had not been associated with cells. So were you able to go back and understand mechanistically how those genes are influencing the mechanical properties of the cell wall?
So recently I have found that there is a gene called DAD-A which synthesizes D-amino acids. D-amino acids are the precursor of these cell walls. So earlier nobody thought that these synthesis genes will contribute to the stiffness, but now we have shown that this particular gene, which makes the precursor, affects the stiffness. Now we are doing more biochemical experiments to study what happens if we overexpress these genes, meaning that we have more precursors. Does it make more stiff cell wall, or having more precursor will decrease the stiffness of cell wall? So we are doing those biochemistry experiments to understand the pathway. So I think what I have done here is I have generated a bunch of genes which next graduate students can come and pick and study and come up with their own hypothesis to test the contribution of these genes in stiffness.
Last chance for a question?
Have you looked at pairwise deletions? So, what if there are
Yeah. So the third point is, chemical genomics, synthetic lethal pairs. So we are combining two mutations at a time, and we are seeing if the combination is additive or they are not additive. So we are doing those experiments. But first we need to make sure that the mutation is affecting the stiffness and how it is affecting the stiffness. Then we can combine and study further, but that is on our future directions list. Yeah.
Okay, so please join me in thanking Rishi. Okay, the next presentation will be by Benjamin Ortiz